High-performance liquid chromatography, or high-pressure liquid chromatography, is one of the techniques in the family of liquid chromatography. Other liquid chromatography techniques include fast protein liquid chromatography, thin layer chromatography, and regular column liquid chromatography that uses gravity to push the mobile phase. HPLC, as its name implies, is differentiated from the others mainly by the high pressure at which it is typically performed. Due to the high pressure involved, at times up to 1,000 times atmospheric pressure, HPLC is usually not used for the separation and purification of biomacromolecules like proteins, but it is often used to separate small biomolecules and peptides in biological samples. On this slide, you can see a detailed illustration of the anatomy of an HPLC system, which is commonly used to separate, identify, and quantify various components in a mixture. First, let's look at the fluid circulation. The solvent reservoirs hold two different solvents, solvent A and solvent B. The degasser removes any dissolved gases from the solvents to prevent bubble formation, which could also affect the separation process. The solvent A and solvent B pumps work together to deliver a precise flow of the solvents to the mixer, where they're combined in a desired ratio. The high pressure pump that ensures that the mixed solvent is driven through the system at a constant flow rate, usually under high pressure. A purging pump is used to purge the system when necessary, such as when changing solvents or cleaning the system. Next is the sample injector, where a precise volume of the sample is introduced into the system. The manipulator enables the selection of specific samples to be fractioned. The switching valve directs the flow of the sample and solvents through the system. The heart of the HPLC is the separatory column, where the separation of components in the mixture occurs. As the sample travels through the column, the different components interact with the column packing material, causing them to elute at different times. Following the column, we have the detector, usually a UV vis detector, which measures the absorbance of the eluting components as they pass through. This generates a chromatogram that helps us identify and quantify the components. Finally, the fraction collector collects the separated components in individual vials for further analysis or purification. The wasteline collects any unused solvents and sample for proper disposal. The beauty of HPLC lies in its ability to efficiently separate, identify, and quantify components in complex mixtures, making it an invaluable tool in various scientific needs fields. High-pressure liquid chromatography is a process of separating components in a liquid mixture. A liquid sample is injected into a stream of solvent. This is the mobile phase, flowing through a column packed with a separation medium, which is the stationary phase. Sample components separate from one another by a process of differential migration as they flow through the column. As bands emerge from the column, flow carries them to one or more detectors, which delivers a voltage response as a function of time. This is called a chromatogram. For each peak, the time at which it emerges identifies the sample constituent with respect to a standard. The peak's area represents the quantity. Let's take a closer look at some of its key components. At the top of the machine, you'll notice the UV vis detector. This crucial component measures the absorbance of light by the eluting sample components at specific wavelengths, allowing us to identify and quantify them. The middle section of the HPLC machine houses the autosampler, which is divided into two modules. The autosampler simplifies and automates the process of introducing precise sample volumes into the system, ensuring consistency and accuracy in the analysis. Finally, at the lower level of the HPLC machine, we have the pump. This component is responsible for delivering the mobile phase, a mixture of solvents at a constant flow rate and under high pressure. The pump's efficient operation is critical for achieving optimal separation and reproducibility in the HPLC process. In this side view of the HPLC machine, we can observe additional important components that contribute to the system's functionality. First, you can see the syringe pump, which is responsible for precisely injecting the sample into the system. The syringe pump works in conjunction with the autosampler to ensure an accurate and reproducible sample introduction. Next, we have the column, which is the heart of the HPLC system. This is where the actual separation of the mixture's components take place. Here we have an example chromatogram illustrating the separation of a sample containing phenol, benzoic acid, and p-toluic acid. Two different mobile phase compositions have been used, buffer methanol and buffer acetonitrile. As you can see, the choice of mobile phase plays a critical role in the elution selectivity of the compounds. Methanol, being a more polar solvent, generally results in a faster elution of polar compounds. In contrast, acetonitrile, being less polar, tends to retain polar compounds for a longer time, allowing for better separation of closely eluting peaks. By comparing the elution times of the three compounds under the two mobile phase compositions, we can observe the effect of the solvents on the separation efficiency and selectivity of the HPLC process. This figure shows a traditional HPLC column packed with porous silica particles. These particles serve as the stationary phase in the separation process. Their porous structure provides a high surface area, allowing for increased interaction between the sample components and the stationary phase. This interaction leads to the differential retention of the compounds and ultimately their separation. The choice of the stationary phase and its particle size can greatly affect the efficiency, resolution, and speed of the separation process. Smaller molecules often provide higher efficiency and better resolution, albeit at the cost of increased back pressure. Now let's take a moment to delve into the chemistry of bonded phases in HPLC columns. Bonded phase packing media begin with a bare silica surface, which is chemically modified through the attachment of the primary bonded phase. Manufacturers express the extent of bonded phase coverage in terms of the percentage of carbon load. Despite the bonding process, some silica remains unreacted, leaving exposed surface silanol groups. To address this, a secondary bonding step is often employed, introducing smaller molecules between the primary bonded phase strands. End capping can be performed using either polar or nonpolar molecules. It is important to note that different columns with the same bonded phase type can still differ in their silanol exposure in end capping. This results in a range of polarities and separating abilities, offering scientists various options to optimize their separation process based on their specific needs and requirements. Here we have a flowchart that outlines the column chemistry choices based on sample solubility. The first step is determining if the sample is soluble in water or polar organic solvents or in organic solvents. This helps you decide between reverse phased or normal phase chromatography. Next, consider the characteristics of the analytes in your sample, such as ionic, polar, natural, or mixed properties. Depending on these attributes, you may need ion pair suppression or specific column chemistries to suit acidic, basic, hydrophobic, or hydrophilic compounds. Finally, choose the appropriate column for your analysis. Some common column types include ion exchange columns like SCX or SAX, and reversed phase columns like C18, C8, C4, C2, high AQ, amino, cyano, or PFP. Here we have an image of the BDS Hypersil C18 column, which is 100 millimeters by 4.6 millimeters in size. This is a reversed phase column, meaning that it is designed to separate compounds based on their hydrophobicity. The term BDS stands for base deactivated silica, which refers to the treatment of the silica surface to minimize unwanted interactions with the analytes. Let's explore the base deactivation process of the BDS Hypersil C18 column in more detail. Base deactivation involves the treatment of the silica surface with a silylating reagent to reduce the activity of residual silanol groups. This process minimizes undesirable secondary interactions, such as tailing or peak broadening, thus improving column performance and enhancing the resolution and reproducibility of the separation. This slide showcases the chemical structures of caffeine, aspartame, and benzoic acid. These compounds are often analyzed using HPLC due to their prevalence in food and beverages. By understanding their chemical structures and properties, we can select the appropriate column chemistry and separation conditions to achieve optimal results in our HPLC analysis. Here we discuss the importance of serial dilutions in HPLC analysis. In our caffeine study, we performed serial dilutions of diet sodas to determine their caffeine concentration. By creating a series of diluted samples, we can establish a calibration curve relative to the HPLC signal response to the actual caffeine concentrations in the diet sodas. This enables us to accurately quantify the amount of caffeine present in these beverages. Next, we prepared a 500 milliliter solution of 50% methanol for HPLC analysis. To make this solution, we combined 250 milliliters of HPLC grade methanol with 250 milliliters of sterile and filtered water. Vacuum filter the solution to ensure sterility and remove any particulates that may interfere with the HPLC system. Keep the solution covered to avoid contamination and label the bottle on the side and the top for easy identification. This is an outline of the process of making caffeine standards at various concentrations. We will prepare solutions at 0 milligrams per milliliter, 0.023 milligrams per milliliter, 0.05 milligrams per milliliter, 0.09 milligrams per milliliter, 0.135 milligrams per milliliter, and 0.180 milligrams per milliliter. In addition, we will degas the soda samples through a filter device to remove dissolved gases that may interfere with the HPLC analysis. Prepare the highest concentrations of caffeine standards first, specifically the 0.180 milligram per milliliter sample and the 0.135 milligram per milliliter solution. From the 0.180 milligram per milliliter solution, we then created the 0.09 milligram per milliliter standard. This approach ensures that the dilutions are accurate and consistent, which is crucial for generating reliable calibration curves and quantifying the caffeine content in diet sodas. The caffeine standards and the soda samples must be degassed by syringe filtration into the HPLC autosampler vials. This helps remove any dissolved gases from the standards, preventing potential interference with the HPLC analysis and ensuring accurate quantification of caffeine content in the diet sodas. Syringe filtration involves passing a liquid sample through a filter device attached to a syringe. This technique removes particulates and dissolved gases from the sample, preventing clogging of the HPLC system and ensuring a more accurate analysis. By carefully transferring the filtered samples into autosampler vials, we minimize the risk of contamination and maintain sample integrity for HPLC analysis. This image is of Diet Coke and Diet Mountain Dew soda cans, the two beverages we used in our experiment to determine their caffeine concentration. By analyzing these popular diet sodas, we can gain insights into their caffeine content and better understand the potential health implications of their consumption. This slide serves to highlight the importance of properly submerging the HPLC lines into a fresh methanol container. Ensuring that the lines are fully submerged in the solution helps prevent air bubbles from entering the system. To minimize contamination risk, cover the top of the container with parafilm. Before starting the analysis, it is essential to purge and flush the system. First, switch on the pump and autosampler to initialize the instruments. The screens will begin a self-test, while the interface and link units should always remain on. To turn on the UV detector, hold down the plus and minus buttons first. Then, simultaneously press down the power button. The unit screen will begin a self-test, ensuring the UV detector is functioning correctly. We will explain the process of purging the HPLC system of air bubbles. Locate the purge valve with the round black knob and a white drip tray beneath it. Use an HPLC syringe and line to purge the system of air bubbles. You must purge lines A, B, C, and D. Adjust the flow rate to 4 milliliters per minute and set the solvent composition to 100% A. Open the purge valve and connect the designated syringe. Pull back the syringe plunger to purge the air from the line until the methanol solvent flows without bubbles. The pressure should read 0 psi while the line is open and being purged. When you close the valve, the pressure should increase to 5,000 psi. Repeat this process for each line, following the corresponding function keys for line B, C, and D. Click Stop when finished. Here we will outline the process of flushing the HPLC system after purging. Once the purge is complete, it's crucial to flush the system to ensure optimal performance. To do this, navigate to the autosampler unit and click the Flush button. Observe the syringe pump on the left side of the autosampler as it begins to work, flushing mobile phase into the waste container. Repeat this flush process 3 times, ensuring that the liquid flushes out each time. After completing these steps, the HPLC system is ready to start running the samples. The syringe pump in the autosampler will pump and flush the mobile phase through the system and into the waste container, maintaining optimal system conditions for accurate analysis. We highlight the importance of setting up the software before running samples on the HPLC system. To begin the setup process, we must first log into the Total Chrome workstation, the software platform that controls the HPLC instrument. Here, we see the main screen of the HPLC software, Total Chrome workstation. Notice sections such as Build Sequence, Report Format, Build Method, and Graph Edit. These sections, along with other buttons, allow you to analyze results, process batches, and review chromatograms. Here we'll focus on creating a method that instructs the software on controlling the instrument and setting up the necessary parameters for our samples. Keep all lines in 50% methanol, purge all pumps, and flush the needles. To run the HPLC instrument, you must create a method parameter and a sequence for the samples. Click the method icon to get started. Select New Method, then click Instrument Selection and choose PEHPLC. Then click OK to proceed. We'll discuss the next steps in setting up a method. Click Documentation and leave it blank. Then click Next. Click Instrument Notes and leave it blank, and click Next again. Adjust the data acquisition data channels to a sample rate of 10 points for caffeine. Set the real-time plot offset to 50 and full scale to 500. Click Next. For the autosampler, set the injection volume to 10 microliters for caffeine with no further changes. For the pump configuration, make no changes. In the pump program, set row 0 to an equilibration time of 1, flow rate of 1 milliliter per minute, and pump A at 100%. In row 1, set the runtime to 5, flow rate to 1 milliliter per minute, and pump A at 100% A. For instrument timed events, make no changes and click Next. We will highlight the process settings. For integration, set bunching factors to 1, noise threshold to 50, and area threshold to 250. Make no further changes. Leave baseline timed events, optional report, and other programs unchanged. In replot settings, remove the option to generate a separate replot. Click Next. Leave global information and LIMS results unchanged. Click Next. For component defaults, set the identification absolute window to 5 seconds and the relative window to 5%. Make no changes to calibration and user values. Click Next. This covers the component settings. For identification, name the component caffeine and set the retention time to 0.169 when known. Leave calibration and user values unchanged. Click Finish to complete the method setup process. Create a report, go to Other, and select Report Format Editor. You may describe the protocol in the documentation window, but this is optional. Click OK, then save the method, sequence, and data in their respective folders. The default report will appear. Click on the words Default Report to change the name to Caffeine Determination Standards. It's good practice to use the same file name for the report as used for the method file and store it in the same directory. The following fields in the report: peak, name, time, area, and height. Delete the rest. Click Report Format Options and select Small. Always use condensed font, print replot with report, and set the area reject to 2000. Click OK, then save and close. Here we describe the process of running samples in the HPLC. First, load the HPLC with the autosampler vials and note the grid location of each sample vial. Create a sequence to label each sample according to its location in the autosampler. allowing your chromatograms to be labeled with the user set title. To run the HPLC, seize control, wait for all icons to be active, and ensure the port and status are both ready. Click Instrument Setup and select the method and sequence from the respective folders. Set the starting row and ending row according to the total number of samples and their location in the auto sampler. Check Start Run when ready, and set the folder location as instructed by your instructor. Click OK to proceed. We have two options to process the results. One involves selecting Batch on the main total Chrome screen, selecting the sequence path for your samples, and ensuring all parameters match the screenshot. The other involves selecting Reprocess Results on the main total Chrome screen and selecting the .rst file of the chromatogram you want to process. Once in Reprocess Results, click Display and then Peak Report. The peak report will provide the peak area information required to create the standard curve graphs. For your lab report, you will need the following chromatographs: the .raw file and the .rst file reprocessed with peak area information. Now you can save the data to share or export. Follow the steps provided to save your raw data and peak report data. Then you can organize the data in PowerPoint, making any necessary edits before including all chromatograms in your lab report's data section. By following these steps, you can efficiently save and transfer your data, ensuring that you have the necessary chromatograms for your lab report's data section. After compiling and analyzing our data, we created a standard curve using the known concentrations of our caffeine standards. This standard curve was plotted in Excel, with the peak areas on the y-axis and the corresponding caffeine concentrations on the x-axis. This curve allowed us to establish a relationship between peak area and caffeine concentration. We then analyzed the soda samples using Hplc and identified the retention times of the caffeine peaks in each sample. By comparing these retention times with those of the caffeine standards, we were able to identify the peak area of caffeine in each soda sample. With the peak area information, we were able to estimate the concentration of caffeine in each soda sample by comparing it to the standard curve. In other words, we used the standard curve to extrapolate the caffeine concentration in the soda samples based on their respective peaks. By leveraging the power of HPLC and standard curves, we were able to accurately determine the caffeine concentration in each soda sample. This knowledge can be invaluable for consumers, researchers, and regulatory bodies alike, as it provides important insights into the composition of popular beverages.